As the sun dips below the horizon, a different kind of light appears. These are the fireflies, nature's living lanterns. Each flash is a silent conversation, a pulse of life in the cool night air. But behind this enchanting display lies a world of incredible biological engineering. This phenomenon, known as bioluminescence, is a cold light produced by a remarkable chemical reaction, all for the purpose of finding a mate. To truly understand the firefly, we must look at its world from three distinct perspectives. First, their adaptability, the story of how they survive and thrive. Second, the physical properties of their light, the science behind the glow. And third, the equipment and methods scientists use to study them. Together, these windows connect the magic we feel with the science we can know, revealing that the more we understand, the more wondrous the firefly becomes. This documentary will be a calm and thoughtful exploration, and we will not rush through the story. Instead, we will take our time, allowing the details to unfold gently, much like the fireflies themselves emerge at dusk. Our narrative will be guided by scientific evidence, drawing from decades of research and observation. We will look at data from scientific studies, explore mathematical models that describe firefly behavior, examine the fundamental physics that governs their light. This is a story built on a foundation of facts, presented in a clear and accessible way for everyone to understand. We will journey through archives to see how our understanding has evolved. We will look at old scientific drawings, compare them with high-resolution digital images we can capture today. Scientific inquiry is a continuous process of discovery, each generation of researchers building upon the work of those who came before. By looking at both historical and modern methods, we gain a deeper appreciation for the persistent human desire to understand the world around us. This calm, evidence-based approach ensures that our wonder is paired with knowledge. The firefly matters for many reasons. Scientifically, its bioluminescence has inspired new technologies in medicine and engineering. The chemical reaction that produces its light is so unique, it has been harnessed by scientists as a tool for research, helping to light up processes inside living cells. Culturally, fireflies are woven into the fabric of our summer memories and folklore, representing a connection to the natural world that is increasingly rare in our busy, brightly lit lives. Their declining populations in many areas serve as a quiet warning about the health of our environment. So join us on this gentle journey into the night. We will travel from the open meadow to the microscopic level, to the wonder of a child's gaze, to the precision of a scientist's measurement. We will embrace a slow, deliberate pace, allowing the story of the firefly to illuminate our understanding. This is an invitation to look closer, to listen to the silent flashes, and to appreciate the intricate engineering that nature has perfected over millions of years. Let us begin our exploration into the world of the living light. Long before a firefly takes to the air, its life begins as a tiny egg in damp soil. From this hatches a larva, a hidden predator living for years underground. Known as a glowworm, its light is a warning to predators that it's an unpalatable meal. After pupating in the earth, it undergoes a profound change. Finally, the adult emerges, ready to take to the night sky and continue the cycle. Once an adult, the firefly's life is a race against time. Its sole mission? Reproduce. To do this, they've evolved a language written in light. Each species has a unique flash pattern, a silent conversation in the dark. The male flies low, broadcasting his call. A receptive female below responds with a precisely timed flash of her own. This luminous back and forth guides the male directly to her, a carefully choreographed dance of light. In certain parts of the world, the firefly's light show evolves into something even more extraordinary. Along the riverbanks of Southeast Asia, thousands of fireflies gather in the same trees. Thailand, Malaysia, isolated pockets of North America. Here, they do not flash their lights as individuals. Instead, they synchronize their flashes. Entire trees pulse with light as one. Sometimes entire stretches of riverbank pulse with light. A unified rhythmic wave of brilliance ebbs and flows in perfect harmony, a natural symphony of light. This phenomenon has long fascinated scientists and observers. For years, it was a mystery how thousands, each with its own internal clock, could coordinate. Some thought it was a trick of the eye. Others suggested a single pacemaker firefly was leading. Research shows its true self-organization. Each firefly is an independent oscillator, influenced by its neighbors. It adjusts its rhythm in response to nearby flashes. Coupled oscillation. Mechanism. A firefly prepares to flash. If it sees neighbors flash just before, it speeds up to match them next beat. If neighbors flash just after, it slows down to join the next round. Over many cycles, these adjustments ripple through the population, syncing them. Mathematical models and simulations predict this emergent behavior without any leader. Why? The evolutionary advantage is debated. One leading hypothesis. It helps the females. In a chaotic sky of thousands of flashes, a female would struggle to pick a single male. By flashing together, males create a shared silence between pulses. During that dark interval, a female's response flash stands out instantly, so synchrony may be cooperative, making female signals easier to see and benefiting everyone. The beautiful language of light used by fireflies is not without its dangers. The very signals designed to bring partners together can be exploited by a cunning predator. In North America, a group of fireflies from the genus Photurus have earned the name femme fatale. These females have evolved a remarkable and deadly form of mimicry. They can imitate the flash response patterns of females from another genus, Photurus photinus. An unsuspecting Photinus male flies through the night calling for a mate. He sees what he believes is the correct, timed response. He has no reason to suspect a trap. The Photurus female has mastered the precise timing of her victim species. The hopeful male follows the deceptive signal, flying closer, continuing the courtship dialogue. He believes he is about to fulfill his life's purpose. But when he lands seeking to mate, he meets a predator. The larger Photurus female grabs him and devours him. Aggressive mimicry, a communication system turned weapon. The reason isn't only a meal. Photinus fireflies possess defensive steroids called lucibuphagens. These chemicals make them toxic and unpalatable. Predators, spiders, predators, birds. Photurus cannot make these compounds themselves. By eating Photinus males, Photurus acquires and stores the chemicals. This makes her, and later her eggs, toxic to predators. She is stealing chemical armor, kleptochemical defense. Some Photinus males now hesitate. They may perform extra verification flashes. In response, Photurus females become better mimics, with whole repertoires of deceptive signals. Sometimes they mimic multiple species in the same habitat. Bioluminescence isn't just romance. It is a complex battlefield where stakes are life and death. The language of light can be used to deceive and to kill. The firefly's ancient language of light evolved against natural darkness. But today, the rapid spread of artificial light creates a profound challenge. This light pollution floods the night, drowning out the subtle flashes crucial for their communication. It's a problem of signal to noise. The male's flash is the signal, and our light is the noise. When the noise is too high, courtship fails, directly impacting reproductive success and pushing local populations toward extinction. Beyond light pollution, fireflies face devastating threats from climate change and habitat loss. Rising temperatures and shifting rainfall disrupt their sensitive life cycles, drying out the soil where their larvae grow. Meanwhile, human development paves over the wet meadows and forests they need to survive. This combination of pressures is testing their adaptability like never before. Their fading light is a somber warning about the health of our shared planet. Section 1. The Engine of Light. Luciferin and luciferase. At the very heart of the firefly's glow lies a precise and elegant chemical reaction. This is the engine of light, powered by two key molecular components. The first is a remarkable substance called luciferin. This is the fuel for the reaction. Its name, derived from the Latin lucem fere, meaning light bringer. Luciferin is a relatively small, heat-stable molecule that holds the potential for light locked within its chemical bonds. It is the substrate upon which the entire process of bioluminescence is built. Without luciferin, there can be no glow. the lantern would remain dark and empty, its purpose unfulfilled. The second critical component is an enzyme called luciferase. While luciferin is the fuel, luciferase is the catalyst, the spark that ignites it. An enzyme is a biological protein that speeds up a chemical reaction without being consumed in the process. Firefly luciferase is a highly specialized enzyme, perfectly shaped to cradle the luciferin molecule. It acts like a biological lock, and luciferin is the key. When the two bind together, the luciferase enzyme initiates a series of intricate steps that will ultimately force the luciferin molecule to release its stored energy not as heat, but as a particle of light, a photon. The relationship between luciferin and luciferase is incredibly specific. The luciferase of one firefly species has a unique structure, meticulously evolved to interact most efficiently with its own species luciferin. This specificity is a hallmark of enzyme-driven reactions throughout the natural world. Think of it as a finely tuned machine, where every part must fit perfectly for it to operate. The enzyme's structure creates a unique microenvironment, a protected pocket where the chemical transformation can occur with extraordinary precision and control, shielding it from other competing reactions within the firefly's cells. This molecular pair is not unique to fireflies, glowing fungi, deep-sea fish, microscopic plankton. However, the firefly system is one of the most studied and best understood. The discovery and isolation of these two molecules by scientists was a monumental step in understanding bioluminescence. It transformed the firefly's magic into a tangible, repeatable chemical process, opening the door for us to explore the fundamental physics and chemistry that allow a living creature to manufacture its own light from scratch. Not all firefly light is the same, with colors ranging from green to yellow-orange. These variations aren't random. They come from the physics inside the luciferase enzyme. The shape of the enzyme's active site determines the energy of the light produced. This is evolutionary tuning. A yellower flash stands out at twilight, while greener light is more visible in deep darkness, a trait optimized for communication in a specific niche. Beyond color, a firefly signal is defined by its intensity. Brighter flashes, created by oxidizing more luciferin at once, can be an honest signal of a male's fitness. But what's truly astonishing is the efficiency. The bioluminescent reaction has a quantum yield near 100%, producing cold light with almost no wasted heat. This is a feat of evolution that human technology still strives to replicate. The concept of cold light is a defining property of bioluminescence, in stark contrast to human-made light, which wastes enormous energy as heat. The firefly bypasses heat entirely. Its light is a process of chemiluminescence, where energy comes directly from a chemical reaction. This allows for a powerful signal with minimal energy and no risk of thermal damage, a tiny beacon of thermodynamic perfection. Beyond color, firefly light has a precise temporal structure, its timing, duration, and frequency. This rhythmic pattern is the syntax of the firefly's language. For example, a Photinus pyrolis male flashes every 5 seconds while flying in a J-shaped pattern. The female responds with a single flash after a precise 2-second delay. These timings are genetically programmed and critical for species recognition. To truly understand the language of fireflies, we must be able to see it clearly because their flashes are incredibly brief, often lasting for just a fraction of a second. To our eyes, a series of quick flickers might blur into a single, indistinct pulse. To overcome this limitation, scientists turn to a powerful tool, the high-speed camera. These remarkable devices can capture thousands of frames per second, far exceeding the capacity of the human eye or standard video cameras. When this footage is played back at a normal speed, time itself seems to stretch. The fleeting flash of a firefly unfolds in exquisite slow motion, revealing details that were previously invisible. With this technology, we can dissect a single flash with incredible precision. We can see exactly when the light begins to ramp up the moment it reaches its peak intensity, and how it fades back into darkness. This allows for the precise measurement of flash duration and the analysis of its shape or pulse profile. For species that produce multiple pulses in quick succession, high-speed cameras reveal the exact interval between each pulse. What might appear as a simple twinkle to a casual observer is revealed to be a structured, complex signal. Beyond analyzing individual flashes, high-speed videography allows researchers to study the movement associated with the light display. For many species, the flash is combined with a specific flight maneuver. The famous J-flash of the Photinus pyralis male is a perfect example. By slowing down the footage, we can trace the exact trajectory of the firefly through the air. We can see how the light turns on at the beginning of the upward swoop and extinguishes at the peak of the J. This synchronization of movement and light is a key part of the signal. The data gathered from these cameras forms the basis for quantitative analysis. Each frame can be digitized, and the light intensity in the region of the firefly's lantern can be measured. This generates a precise dataset of brightness over time, which can be plotted and compared mathematically. Researchers can compare the flash patterns of different individuals, study variations between different populations, or investigate how environmental factors like temperature affect flash timing. The high-speed camera transforms a beautiful natural phenomenon into a rich source of scientific data, allowing us to decode the firefly's language one frame at a time. To understand how the firefly's lantern works, we must venture into a world far too small for our eyes to see. This is the realm of the microscope. By using powerful light microscopes and electron microscopes, scientists can examine the intricate cellular architecture of the light-producing organ. Under high magnification, the lantern is revealed to be a highly organized structure, not just a simple bag of chemicals. It is composed of specialized cells called photocytes, which are packed with the ingredients for light production. These cells are interwoven with a dense network of nerves and tiny air tubes called tracheoles. Electron microscopy provides an even more detailed view, allowing us to see the substructures within a single photocyte. We can visualize the mitochondria, which play a crucial role in controlling oxygen supply. We can see the peroxisomes, the tiny compartments where the bioluminescent reaction is thought to take place. These incredibly detailed images reveal a structure that is perfectly engineered for its function. The close proximity of the air-supplying tracheoles and the energy-providing mitochondria to the light-producing machinery is a testament to the efficiency of the system. Beyond just imaging, modern microscopy is often combined with techniques of microanalysis. For instance, scientists can use fluorescent dyes that bind to specific molecules, like calcium ions or nitric oxide. When the firefly flashes, these dyes light up under the microscope, allowing researchers to visualize the flow of chemical signals within the lantern in real time. This method provides direct evidence for the role of these molecules in controlling the flash. It is like placing a tiny camera inside the cell to watch the machinery of light in action. Furthermore, advanced techniques like mass spectrometry imaging can be used on thin slices of the lantern tissue. This method can identify and map the spatial distribution of different molecules, including luciferin, chemical precursors, byproducts. This allows scientists to see where the fuel is stored, where the reaction occurs, and where the ash of the reaction accumulates. By combining these powerful imaging and analytical tools, researchers can build a complete, multi-scale model of the lantern, from its overall organ structure, down to the precise molecular events that generate a single photon of light. To understand the firefly's lantern, we must venture into a world far too small for our eyes to see. Under high magnification, powerful microscopes reveal a highly organized structure. It's composed of specialized cells called photocytes, interwoven with nerves and tiny air tubes. Using fluorescent dyes, researchers can even watch the flow of chemical signals in real time, revealing the machinery of light in action. While a spectrometer tells us about the color of the light, a different set of tools is needed to measure its brightness. This is the job of photometers and light meters. These instruments are designed to quantify the intensity or luminance of a light source. A simple light meter, often used by photographers, can give a general reading of ambient light levels. For scientific purposes, however, researchers use more sophisticated photometers that are highly sensitive and can measure very rapid changes in brightness. These tools are essential for studying the dynamics of the firefly's flash and the information it conveys. In a laboratory setting, a firefly might be placed in a dark chamber with a photodetector, such as a photomultiplier tube, aimed at its lantern. This setup allows for the most sensitive and accurate measurements. The photodetector converts the incoming photons into an electrical current, with the strength of the current being directly proportional to the intensity of the light. This electrical signal can be recorded and plotted over time, producing a high-resolution graph of the flash pattern. This data allows for precise measurement of peak brightness, flash duration, the intervals between flashes, providing the raw data for analyzing the temporal code. Portable light meters are crucial for fieldwork. They allow researchers to measure the brightness of firefly flashes in their natural habitat and the ambient light conditions in which they are communicating. A scientist can use a light meter to quantify the level of light pollution from a nearby streetlight or to measure the natural decline of light during twilight. By correlating these environmental light levels with firefly activity and flashing behavior, researchers can scientifically test the impact of artificial light on firefly populations. These measurements provide the hard data needed to advocate for conservation strategies, such as shielded lighting. The use of these instruments also allows for comparative studies. Researchers can measure and compare the flash intensities of males, females, and compare the brightness of different species. This can help answer evolutionary questions. Is a brighter flash a signal of a healthier male? Do females preferentially respond to these brighter signals? By transforming the subjective quality of brightness into a hard number, photometers allow scientists to treat it as a variable in their experiments. This quantitative approach is fundamental to the scientific method, enabling objective, repeatable, statistically valid conclusions about the function of light in the firefly world. Our journey into the world of the firefly has illuminated a story of incredible complexity and elegance. We saw how its life unfolds in stages, written in a silent, flashing dialogue that allows mates to find one another across the darkness. We delved into the molecular engine of its cold light, a marvel of efficiency where chemistry becomes a visible glow. And we explored the scientist's toolkit, where automated systems transform fleeting moments into quantifiable data. These windows, adaptability, physics, and equipment, are interconnected. Together, they show how a simple glow is the product of millions of years of evolution, revealed through human curiosity. A meadow full of glowing fireflies. The next time you see a firefly's flash, you may see more than just a flicker. You will be witnessing a conversation. This new understanding invites us to become more mindful guardians of the darkness. We can turn off an unnecessary light, or let a part of our yard grow wild. These small actions can make a real difference, helping these living lanterns thrive once more.